Hello, I'm Jim Cornell, and this is Base to Base, the weekly biotech podcast from Air Coastal Media. Yes, welcome again to the Base to Base biotech podcast, and this is number 45, and it's going out on February the 6th. We made it through January, although having said that, February the 6th is, I was told, Valentine's Shopping Reminder Day, and I couldn't believe it's actually a day, but yes it is. I despair. It's a pretty boring week, all work and all rain and wind. Not sure if there's a connection, but our toilet door fell off. Not that I'm blaming our resident teenager, and we don't have any video inside the house for proof, so it shall remain a mystery, just like who ate the last bagel yesterday. Last week we had a conversation with Pan Cancer Tea CEO Rachel Abbott, and we also talked to Eric Manting, CEO of Mendus, and Jeanette Hoogstad, CEO of Northex Biologics. This week it's two interviews, and the first is with David Bejker, CEO of Affibody, about radioligand therapy and more. And then it's a conversation about chronic pain and urology with Martin Gleave, founder and chief medical officer at Sustained Therapeutics. Just a reminder that if you do have news or suggestions for who should be on the podcast, or indeed any other suggestions or comments, please do get in touch at jim@deeptechdigest.com, which is also the address you can use if you're interested in advertising with us, or sponsoring a podcast episode, or for event coverage, or if you just want to chat, as long as it's about something I understand, so not reality TV. Alright, let's take a look at the news you may have missed over the past week at deeptechdigest.com. And this week I even had some left over for next week. A broken DNA repair tool accelerates aging. Eli Lilly's injectable medicine and device manufacturing facility is going to be built in Pennsylvania. And Trace Biosciences' nerve-specific imaging agent has received FDA IND clearance. A new dementia treatment strategy has been proposed. Feta Pharma is building a new manufacturing site in Germany, and a sarbecovirus vaccine trial has started. There are positive top-line results from an acne study. Researchers in Belgium have developed a more reliable platform to test antibody medicines, and the California Institute for Regenerative Medicine has approved a $100 million plan to accelerate genetic therapies for patients with rare diseases. A mucopolysaccharidosis type 2 treatment has received FDA Rare Pediatric Disease designation. Breakthrough medicine has emerged from stealth to develop cancer drugs, and Johnson & Johnson's prostate cancer drug has cleared a hurdle in Europe. And you can read all of these and plenty more at deeptechdigest.com. Okay, let's move on with the first conversation, and it is with David Bejker, CEO of the Swedish company Affibody. It's a conversation about Affibody molecules and radioligand therapy, or radioligand, depending on your accent. Anyway, rather than me give you a long introduction to the company, we'll leave that to someone far better positioned to do that than me. Hi David, thanks a lot for joining us today from a place even further north than I am. Pleased to have you on the podcast. I wonder if first we could start with a bit of background on Affibody.
Yeah, so Affibody is at heart we're a protein engineering company, but we've moved our platform now to a place where we have more than 1,000 patients dosed. We have long-term safety and efficacy. So we think we kind of established that core platform, the core protein engineering platform from that perspective.
And where is the company headquartered?
We're based out of Stockholm. We're about 60 employees. We're this kind of pure biotech, and we now focus our own internal resources kind of exclusively on radioligand therapies. We do have a ionized partnership portfolios where we have partners with like KSC, Rally Bio, etc. We're working with more kind of I&I products.
Okay, great. I guess it would be helpful maybe to give a bit of background and explanation as to what the field of radioligand therapies involves.
Yeah, so I mean, I think it's really cool with radioligand therapy because if you look at like the AACR's pillars of cancer treatments, there's 5 pillars. You have surgery, you have radiotherapy, you have chemotherapy, you have molecularly targeted therapies, and you have immunotherapies. So we know cutting cancer out works. We know radiating cancers from the outside kind of works. So we have this kind of molecular targeted therapies coming. So what if you actually combine targeting with radio? So that's what a radioligand is. It tries to take what used to be external beam radiation or still is external beam radiation. You put the radioactive isotope onto a carrier. That carrier is molecularly guided to a certain cancer target, and then you can kind of kill the cancer. So that's the idea.
All right, that's a great overview. Thank you. So maybe we could turn to your company now, and maybe you could give me a bit of information on your Affibody platform and how that works, what that does.
Yeah, so the Affibody platform is, I mean, the word we and others kind of use to describe platforms like this is mini proteins. So you can either think of the Affibody molecules as large peptides with the structures, or you can think of them as very small proteins. So an Affibody molecules is about 6.5 kilodaltons in molecular weight, or 58 amino acids. So that's kind of the size of it. And that's a perfect size for and a perfect biodistribution for radioligand therapies.
What are some of the challenges that maybe radioligand therapy can address that others can't, either better or different to other therapies?
Yes, I think what we've seen in the radioligand space is that successes has been small molecules and peptides. There are really like 2 huge successes, Pluvicto and Lutathera. And if you look at the pipeline of small molecules and peptides, actually 85% of that pipeline targets the same target as those 2 successes, PSMA and SSTR2. And if I were to include FAPI and DLL3, that would cover almost the entire pipeline. So that's kind of where the industry did stand a couple of years ago, I should say. And if we take RLTs as an analogy to ADCs, with ADCs, there's never been a problem in raising the antibodies, the A. I mean, you can get antibodies to any type of targets you really want to. It's been linkers and all of that stuff that's been difficult. But antibodies aren't that great for radioligands. So what we're trying to solve is broadening the target space, new targets, so that we can actually broaden radioligand therapies to other indications and new patients. So that's kind of where we play.
There's been quite a few deals in the field using traditional peptide antibody approaches. How's your technology in advance or different to current precision oncology?
So I think that the answer is to a large extent this fact that we can go after targets that have been difficult to target with peptides and small molecules, and there's quite a few of those. And we have the ability to generate like a really high affinity. We're talking picomolar or even sub-picomolar affinities with our binders, which is kind of an antibody-ish feature that's been proven difficult for peptides and small molecules. And of course, stability, engineerability, and all of this is important in the RLT space as well.
So when it comes down to the specifics, could you give me a little bit more detail on ABY271, what that is, how that works, those kind of things?
ABY271 is our lead radioligand program. It's our lead RLT. That's a molecule targeting the kind of well-established and well-known oncogene HER2. We're coupling our molecule with lutetium-177 as a therapeutic payload to kill off the cancer cells. That's a beta emitter. And we're using that in patients that metastatic breast cancer patients that are known to have a lot of HER2 expression. We're using that therapy. So what's interesting is the fact that HER2 is a pretty well established target. You find naked antibodies, combination of antibodies, you find multiple ADCs approved, but there isn't any real competitions for radioligands. It's just us and another company that's actually in phase 1 clinical trials.
There's a lot of companies, as you said, concentrating on HER2. Do you see your treatment as being a standalone treatment or in use with other therapies?
So I think that's a great question, because we see, of course, we have to start on the standalone route when we develop, but we have designed our molecule so that it's not, the binding surface of our molecule does not interfere with the binding surface of the antibodies that are out there, trastuzumab and pertuzumab. So actually we see it as a standalone, but with the optionality of combinations, and combinations with radio is something that's really, really exciting. We even have pretty nice animal data combining our 271 with just Herceptin that shows some synergistic benefit, or at least additive benefit, I should say.
Are there any challenges associated with radioligand therapy at all? I mean, for example, are there any potential side effects?
Yeah, I mean, I think that if I start on the side effect question, I think that's the interesting thing with radioligands, right? The side effect profile is really quite benign. And I mean, and the most advanced, what you see here is that Pluvicto, the PSMA targeting from Novartis, is now actually approved ahead of chemo because of the nice side effect profile. So radiation is pretty benign from that perspective. The issue with the radioligands is kind of, you have to get the isotope to the hospital with the exact activity of becquerels or giga or megabecquerels that the patient is going to be injected. So it's a kind of a just-in-time delivery. So the logistics and the distribution of that, and to some, in certain cases, the patient handling is what's the challenge.
Okay, when it comes to clinical trials, could you tell me where you're at along that particular pathway?
Yeah, so we're now in the first in human with 271. We just had the first readout. So the idea here is to establish safety and dosimetry. And dosimetry is this concept in our world where you can calculate how much radiation goes to different organs and to the tumor. So you can kind of dose plan from that perspective. So, and we're moving into the second part of that study now, which kind of means that we're starting to dose escalate the radioactive dose. And after this part has been done, the plan is to next year to start a proof of concept study. So a traditional POC.
All right. So could you tell me a little bit about the TRC decision, what that is or what that was, and what the implications of that are for the company going forward?
So we started this trial in the fourth quarter of last year, in 2025, and just before Christmas, we had our first trial review committee meeting, which really is a safety committee to a large extent, because this is a first in human, so it's a safety focus. And what they said is that they look at the first two patients and said, well, we had images for the first two patients at 0.5 gigabecquerel, which is a very low dose for lutetium SPECT. They said, well, this looks like the biodistribution you were gunning for, very limited uptake in normal organs, and by that we mean liver, kidney, and bone marrow, which means that you can go on and escalate the dose. We also saw targeting of tumor in patients here, but that was kind of less of the important aspect. The important aspect was that the biodistribution from mice to man translated very well, so we can move in with higher doses and patients that are actually on progressive disease, because these first two patients were what I tend to call healthy patients, well controlled, no disease progression.
So ultimately, what do you hope or expect from 271 when it comes to the end patient and what the end result is?
So I think what we hope for here is to put an alternative forward. Because the sad thing about metastatic breast cancer is that although there's been a lot of innovation, a lot of new drugs happening over the last 20-25 years, the number of women that die from metastatic breast cancer has remained constant over this period. So what we see is that drugs that actually work quite nicely prolong survival, but they're not cures, which means that we want to provide another option, something that's, I mean, we tend to use sometimes the word orthogonal, but we have a orthogonal efficacy, meaning our efficacy is driven by the radioisotopes and the DNA breaks they cause, and a different adverse events profile, the radioactive, rather than chemotherapy or ADC chemotherapy, so to speak.
So the lead candidate 271, does that have implications or uses with other things, for example, other solid tumors?
Yeah, absolutely. I think there is gastric cancers are, of course, important. And we should also note that the lead ADC and HER2 is now approved for any HER2 expressing cancer. And there are several cancers that do have HER2 expression, maybe not as large as metastatic breast, but you would have lung, you would have urology, bladder cancers, you would have actually colorectal. So there's a whole host of them.
All right. Do you anticipate tackling those in the future? Obviously, you're busy with what you're doing already, but.
Yeah, so no, I mean, our plan kind of in the clinic is metastatic breast first with high HER2 expression. Then we want to move into low HER2 expression where we know from the ADCs that it works. So that's actually a pretty big chunk of patients. And then our next after that is gastric. So that's kind of the plan forward.
Right. You're clearly extremely busy with everything that you're doing now, but do you have other things in the pipeline that you're working on either now or potentially in the future?
Yeah, so we have our second radioligand coming up here, which is a drug we call 071 that's targeting B7H3, which is a oncogene or oncologist target, I should say, that is starting to be established. There are some nice ADC data coming. This is a pan tumor target as well, with lung cancer, prostate, triple negative breast, and things like that. So that we made a candidate drug selection last year, meaning that we have kind of the animal biodistribution, animal therapy studies, and this year we're spending to kind of get it ready for the clinic, and the plan is to start that clinic next year as well.
All right, so looking ahead, what does 2026 look like for Affibody? What are you looking forward to?
So 26, dose escalation in the HER2 study, of course, super important, getting all that safety and dosimetry data in place to be able to start a proof of concept study. Secondly, of course, getting 071 ready for the clinic to make that move. And of course, with the dose escalation, I should say there'll be chances to present data and get that out in the public domain and for physicians and patients to know about.
I guess it's pretty much everywhere right now and in pretty much every conversation, but has artificial intelligence played a role in the things that you're doing at Affibody?
Yeah, so not for 271, I couldn't honestly say that, but for 071, yes. Because 271 was kind of ready before there were really good tools. But for 071, we have actually, when we looked at linker designs and amino acid designs for that, we did spend time with artificial intelligence tools. And for the drugs that are coming up after 071, so to speak, it's kind of a key ingredient in our process nowadays. It's interesting because it's just, the fancy word is lab in the loop, but my way of thinking about it is that these artificial intelligence models that we use and employ, it's just like another lab instrument. You get this boost of understanding of predictability.
If we can just go back to the overall big picture of radioligand therapy, how do you see the future of that, and how do you see that evolving over the years?
So, I think I'll take the 2026 perspective. I think this is going to be an exciting year. We have a lot of companies that are kind of pure play biotechs like ours. We have us, we have Actis, and we have ArtBio, just A, and there's others on A, Alpha 9, etc. But I think a lot of us are now kind of in this space where we're starting trials with innovative approaches. So those will kind of read out. ArtBio started their trial pretty similar time as us. So Actis is maybe a little bit ahead, but in first in humans as well. We'll get a lot of data on new targets and new innovative approaches. So that's going to be super exciting, because at the same time we saw in 2025 that this move from pharma that you said, with the deals and all of that, we're moving into product focus with new targets, and maybe the ACP3 deal between BMS and Phylochem, $350 million upfront, is kind of the signature piece for that last year. I think we'll see that continue, target-based deals or product-based deals. And, it's going to be exciting to see what this fact that Pluvicto can now be used ahead of chemo, what that actually will mean for the entire field, because that's a big kind of selling point for RLTs over ADCs.
You mentioned some of the company names, and clearly there are a lot more. Does that mean that the space is very crowded because of that?
I would say the opposite. This is actually a space where I can almost keep track of all the names of the companies, which, if you compare that to ADCs, that's virtually impossible. There is a scarcity of companies, and there's a scarcity of targeting agents that work for radioligands. Of course, as I just said, a lot of us are just early in the clinic, so maybe a couple of the approaches won't pan out, but we're now at least kind of exploring that as an industry.
All right, thanks so much. Those are really the only questions that I had for you today. Is there anything else that we didn't cover that you wanted to mention?
So maybe what I could mention is that we put out a press release that we're doing kind of this smaller race to get led by our lead owner, which kind of funds the entire thing that I've been talking about here, the 271, getting that ready for proof of concept, getting the 071 into the clinic, which puts us in a really good position as well. I was going to mention, actually, our partnerships, and is fundraising important to Affibody? Partnerships has always and will always be important for us as a company. And the concept is we can build a lot more Affibody molecules than we can ever push forward. So right now we've said that I&I is exclusively done via partnerships. Radio, we want to take to a place where we think we have established it. We're getting there. We're clearly getting there. And then, of course, see if we can extract more value for us and for patients via partnership, then we'll go down that route. Well, a really good overview of radioligand therapy and Affibody there. Radioligand therapy is definitely a useful tool in the fight against cancer, among other things, and the more tools the better, that's for sure. From Sweden we're going to another country that copes very well with winter, and that is Canada. Sustained Therapeutics is a spin-out from the University of British Columbia on the west coast of the country. Having spent most of my adult life in Canada, although most of it in the east, it was great to talk Canada and hockey and other things with Martin Gleave, founder and chief medical officer at Sustained Therapeutics. Of course, at some point we did turn our attention to life sciences. So, thanks for joining us today, Martin. Looking forward to hearing all about the company and its work. Maybe we could start with some details about the company itself.
Sustained Therapeutics is a startup from the University of British Columbia, from my laboratory or center. I just stepped down as director of the Vancouver Prostate Center, director of the Institute of Urologic Sciences that now contains a number of centers that includes the Vancouver Prostate Center. It's a large translational cancer research enterprise with 25 scientists, about 250 people. And in that space, we do a lot of discovery science, create a number of different patents that we then ultimately will either out-license or spin off into biotech companies. The work around Sustained Therapeutics started to try to create a series of formulations for intratumoral injections, various cancers, notably prostate cancer, where we could inject the cancer with a number of different anti-cancer agents. And while that worked in animal models, preclinical studies, as we thought about how to develop it, we recognized that there was a lot of unprecedented space that would make that a real challenge to finance down the road. So we took our formulations, which have become now IP protected, and looked for other areas of unmet clinical need where we could inject a active drug into a particular area and deal with a clinical problem. And so one of the lead compounds was reformulating what we call STO1, which is a polymer, liquid at room temperature, but once you inject it into an aqueous solution into the body, it forms a waxy soft solid that then hydrolyzes and releases its payload. So we chose to go forward with lidocaine for areas of chronic pain as one indication. There are several others which we can touch on with drug products as well in clinical development. But the lead now is the STO1 formulation of lidocaine. And we thought about areas of chronic pain, what we call chronic neuropraxic pain. These are chronic pain syndromes where the signals along various nerve bundles are disordered or disrupted and signal pain back to the person. These include things like chronic pelvic pain, trigeminal neuralgia, post-amputee pain, so on and so forth. But as a urologist, I was most familiar with chronic pelvic pain as a problem, and in particular, a subtype of chronic pelvic pain called chronic scrotal pain, which mediates down through the spermatic cord into the scrotum. And we diagnosed that by injecting it with lidocaine, just to diagnose it as the localized problem of nerve pain mediation. And so we thought, well, if we could control that pain for 2 hours with just simple lidocaine, if we could inoculate with a lidocaine formulation that had 4 weeks of activity, then we had a clinically viable therapeutic. So that was the main impetus for spinning off that product from the University of British Columbia into Sustained Therapeutics.
I know that it sounds pretty obvious when you're talking about chronic scrotal pain, but could you give me a bit more background on what that is and how frequent that is and whether it's associated with specific conditions or whether it can be a standalone condition?
It is a standalone condition. Obviously, scrotal pain can be caused by infections, epididymal cysts, or infections, epididymitis, but those are relatively simple to diagnose and treat and not chronic. Chronic scrotal pain is, as I said, mediated through the spermatic cord and is associated with no distinct pathology in the scrotum itself. Its incidence is not, that rare. It's harder to capture only because it's, not something that people want to share or talk about. I would say that if you go to somebody's house and talk about your chronic scrotal pain over dinner, you're not going to get invited back for dinner. So it's not something that people want to talk about. It's estimated that, when you look at the market for chronic scrotal pain across the North American market, it's about a $300 million a year market for a drug product like this. Right now, it's managed by the use of lidocaine as an injection to diagnose it. It's then managed by anti-inflammatories. You want to avoid the use of opioids, obviously, because of the risk of addiction. And things like gabapentin are also used. And ultimately, if those fail, then you go on to surgery and do what's called denervation surgery, where you basically devascularize or deinnervate the spermatic cord. And that's associated obviously with surgery, but also the risk for testicular atrophy and other issues in the scrotum. So surgery becomes the sort of end treatment, but it's something that you want to try to avoid if at all possible.
Is this an area that there are a lot of companies working in? It doesn't seem like one that you get a lot of press releases about.
And so we chose this development path because this spin-off is a urologic company. We understand the space. We understand, I think, the ways that you can design a clinical trial to optimally select the patient for the trial, and we can get into that from a protocol development perspective. But it also represents a foothold. So if you move in and get approval into a smaller niche area for chronic scrotal pain, our next plans are phase 2 trials in chronic pelvic pain, which is a much larger problem. It spans both men and women. It's also called chronic prostatitis syndrome in men, but it isn't a prostatitis problem. Again, it's a neuropraxic condition mediated by the pudendal nerve. Similarity in women, chronic pelvic pain via the pudendal nerve. And there are many interventions in pain clinics, so to speak, that aim towards injecting the pudendal nerve with long-acting anesthetic agents to help control that pain. So while chronic scrotal pain becomes the first indication to gain a foothold from a regulatory perspective, the goal for the product is to move it into chronic pelvic pain, alongside that to move it into things like post-amputee pain and trigeminal neuralgia pain, and ultimately other neuropraxic conditions, including spastic conditions associated with cerebrovascular accidents, so on and so forth. So there you can inject the motor nerve to help to relax the spastic muscles and help with physio and rehabilitation. So there are many other indications downstream, but as a small biotech company, you have to stay focused on your lead compound, lead indication, and get that approved in order to then be able to finance the other indications down the road.
Right. Speaking of that lead indication, could you tell me a little bit more about STO1 and how that works?
Yeah, STO1 is based upon a polymer of PEG and PLGA at different unique lengths that allowed us to get IP protection around it, that provides a property whereby it's relatively low viscosity in a non-aqueous solution at room temperature that allows it to be injected through a needle that, say an 18 gauge needle, a 20 gauge needle. It's formulated with lidocaine at 2 different concentrations right now, 70 and 140 milligrams. And we inoculate up to 3 cc's into the cord to deposit the payload in the stromatic cord. So essentially, it's a polymer that is semi-viscous. Once injected into an aqueous formulation, the water itself interacts with the polymer solution, allows it to form a waxy paste that remains immobilized at the site of inoculation. And then through the process of hydrolysis, breaks down the polymer. As the polymer breaks down, it releases its payload into the local area. And it releases this over a 3 to 4 week period of time. It does allow for repeat injections as well, which is part of our randomized phase 2 trial that just completed.
Right. Yeah. Speaking of which, could you tell me a little bit about the recent results from that phase 2 trial?
As we developed STO1, we initially did a classic dose escalating 3+3 design phase 1 trial that allowed us to show safety, feasibility, and select our phase 2 volume and dose, which ultimately was 3 cc's. We wanted to look at both 70 and 140 milligrams per ml. We wanted to maximally load the paste up to 140 milligrams, thinking that we would want to deliver as much lidocaine as we could safely. The 70 milligram was more around the optimal loading concentration per the polymer. The trial enrolled 63 men across Canada. It was a multi-center randomized phase 2 trial, single blinded. So the patient was blinded to the therapy that he was receiving. As part of the screening process, the patient had an injection of lidocaine into the spermatic cord to document at least a 2-point decrease in the NRS pain score. And if that happened, then he was eligible for the treatment. We didn't want to enroll people who obviously was not going to respond to the drug because there are other syndromes of pain that are not going to respond to a local injection. So those patients were excluded from the trial. In addition, during the screening period, the patient had to complete a NRS pain score and demonstrate a greater than 4 pain score over a 1-week period of time. So the inclusion criteria included men with chronic scrotal pain, NRS pain score greater than 4, with a demonstrated response to single-dose lidocaine. Those patients were then enrolled, randomized to one of 3 arms. Standard of care was lidocaine alone. That was in one arm. The other 2 arms was standard of care lidocaine plus either 3 cc's of STO1, 70 milligrams, or 3 cc's STO1, 140 milligrams. And it, again, enrolled over time across the country. And just recently, again, we completed accrual, and our primary efficacy endpoint was reached. The primary efficacy endpoint is a patient-reported decrease in the NRS pain score of greater than 2 at days 56 post first injection. So patients received an injection, obviously, at day 0. They were able to receive another one at day 28 and again at day 56. And day 56 was the endpoint for essentially 2 inoculates, right? Day 0 and day 28. And if patients felt they were receiving a benefit, they could go on to get repeat injections again at day 56, cycle 3 and cycle 4 as an example. So we had a per protocol statistically significant response rate in the 70 milligram per milliliter arm, and P less than 0.05. I don't want to get into the exact numbers yet because this is going to be presented at the American Urological Association meeting in a few months. And I just want to stick to the press release numbers and say that, we were very pleased. The primary efficacy endpoint was met with robust statistical power. There was a strong trend of benefit in the 140 milligram arm, but it was not as great as a 70 milligram arm. The other aspect of the study was that in patients in the standard of care arm, after they met or crossed over day 56 and had their primary efficacy endpoint evaluated, they could be crossed over to the STO1 arm, either 70 or 140 milligram. And we had the majority of crossover patients from standard of care actually achieve a response in the crossover population as well. So I think that overall, the response rate data met our expectations, and they met, a statistical significant endpoint that allows us to now justify moving into phase 3 from a registration path.
So what are the steps to getting that phase 3 underway?
It will be a 2-arm trial going forward with 70 milligrams. And we anticipate a sample size of about 120 patients, 60 per arm, similarly with crossover in the standard of care arm. And with, relatively, I think this is one of the things that Sustained Therapeutics can leverage from, is that the development path is relatively short and the sample size calculations are relatively small to allow us to achieve a potential, regulatory endpoint for an NDA with another randomized phase 2 120 patient trial that will open across North America, essentially Canada and the US down the road.
Now that you're post phase 2, I guess, are you looking to more financing, partnerships, that kind of thing?
While we remain relatively plastic around ways to move this forward, our plan is to, number one, out-license STO1 to other jurisdictions for development, China as an example, Europe as another example. We're going to stay in the North American space. Those opportunities are being explored and discussed now. But for the North American space, our plan is to move forward with a Series A and push this across the finish line ourselves. I mean, we are a relatively small, lean company, but we have a great experience both in the commercial development of assets, but also the clinical development of our products. In addition to STO1, we have another product, STO2, in randomized phase 2 trials across Canada now. This is a different polymer, more of a mucopolysaccharide or mucoadhesive formulation with gemcitabine for upper tract urothelial cancers. So that's in phase 2 trials. And we have another product that will be in phase 2 trials, already Health Canada approved, IRB approved at the institutions, for a product in erectile dysfunction, again, using the mucoadhesive formulation inserted at the tip of the penis to help men with erectile dysfunction going forward, which is obviously a huge market as well. So it just illustrates that these formulations that we are IP protected for have the ability to take on different payloads and target areas of unmet need that allow us to develop these in a time and capital efficient manner.
Right. And as you mentioned, small company, you don't want to completely overextend yourself by doing too much. But what does the future hold, do you think, for the company? Where do you go from here?
And we hope, our vision is to bring this across the finish line. We have a team of experienced people who have commercialized products before and are in a position to, if the phase 3 trial is positive, then seek to commercialize it across the North American space. That may involve either us doing that and building out the company or licensing it to another commercial entity down the road. How we get there, whether or not it's through a Series A and venture capital or through an IPO, will determine things in the environment that we can't predict right now. But ultimately, we see ourselves taking this downstream to an NDA anyways.
All right. Are there a lot of spin-outs coming out of UBC?
I mean, UBC is number one in Canada for biotech spin-offs. Companies like Angiotech, QLT, Zymeworks, Abcelera, the lipid nanoparticle that helped commercialize the COVID vaccines of Pfizer and Moderna, that IP is out of UBC. And that similarly now is being applied to a lot of RNA-based therapeutics in the cancer world and elsewhere. So UBC is certainly one of the top 10 universities in North America for IP generation and biotech spin-offs.
Another really interesting conversation and lots of work in several different and important areas. I read in the news this morning, and I'm assuming it's not fake news, that the UK government says 75% of people will survive cancer by 2035. That is great news. Now we just need to get that to 100% and hopefully sooner. But there do seem to be a lot of breakthroughs, which is definitely excellent to hear. And let's hope that that also can be extended to all countries around the world, not just one or two. So that's it for this week's podcast. Still lots of emails coming in with both news and requests to be on the podcast, which is good. I guess it's a sign of the times, but lots of proposals for guests in the AI and data field, which is great. But then we can't really have every episode be on the same subject. So it is a challenge sometimes to be balanced. We haven't yet had a request for the guest to actually be an AI chatbot. That might be interesting, as long as they don't try and take over as host of the podcast. I did watch a baseball game from Asia a few months ago, and the play-by-play was 2 AI voices. It was about 10 minutes before I had to mute it. It was just awful in so many ways. Anyway, if you do want to get in touch with guest opportunities, or if you want to advertise, please do get in touch at jim@deeptechdigest.com. So, until next time, wherever in the world you are, I hope you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Base to Base Biotech podcast. The Base to Base Biotech podcast is an Air Coastal Media Limited production.